Thank you very much. Donc, I have the honor to introduce Michael Kubist. Michael Kubist is head of the laboratory of Gene Expression Laboratory Institute of Biotechnology in Czech Academy of Science in Czech Republic, but also co-founded the Tata Biocenter. And I have the honor because Michael contributed to the development of qPCR, introduced the qPCR for single-cell gene expression analysis. So today present our work on spatiotemporal characterization of wound healing and regeneration in amphibians and of ischemic brain injury in mice. Thank you, Mike.
So this work has been mainly performed by Radek Šindelka and Lukáš Valihrach in the group that I'm heading at Czech Republic. They're both senior researchers, and a small part of their team is shown there, and Daniel Zucha is the student who has done most of the work. Oh, super. You got a glimpse. That graph shows, thank you. I'll try again. No. Yeah. So thank you. So this graph here shows the increase in single-cell publications over time, and I think that everybody somehow is involved in single-cell expression profiling or other studies. Note that this graph actually starts in 2007. So actually our team published the first paper in 2005. That was the first single-cell paper, and I'm going to show just a few slides from that very, very old paper because it's 18 years ago now, reflecting the experience that our team in Czech Republic has gathered. And we were working at that time together with researchers at Sahlgrenska University Hospital in Gothenburg, Sweden. And essentially the challenge that we were looking at was the islet of Langerhans that you can see here. They are stained for different cell types, beta cells, alpha cells, and delta cells. And essentially you can see that tissues is a mosaic of different cell types. We knew that, or today everybody knows that. But in those days it was very, very hard to study a particular cell type because there was really no way how to do it. So we managed to either collect the content of the individual cells directly from the islets or using patch clamp technology or dissociated. And then we measured expression profiles of the different cells, and the exciting discovery in 2005, which today is established, of course, is that there was a large heterogeneity even across like cells. In fact, this is even a cell line, these data on Min6 cell line, which is an measuring insulin 1. And doing some statistic on that, we could see that most of the expression actually originates in very, very few cells. So although the cells are the same type, some cells contain most of the transcripts. And if we just change the scale of that graph into log scale, we could show that the distribution of transcripts across different cells of the same type is log normal. So we have a normal distribution when we are in logarithmic scale. And this has turned out to be essentially an underlying concept of gene expression in like cells. Today we know it's not formally a log normal distribution. It's a little more advanced, like a gamma distribution, but it approximates very well. And and as a consequence, if you are interested in the typical cell in your population, then you actually see that if you calculate the average expression in a linear scale, you're not actually getting the expression of the mean cell, the cell in the middle. Because if you want to look at that, you have to take the average in log scale, which is something you have to calculate from single cell data. You can't do that from global data. And our first paper was summarized in Nature with the interesting title of Which Mean Do You Mean? So this is in 2005 when this field started. And I also want to mention another paper that we did many, many years ago in 2007. Again, this was the start of that graph. And that is when we pioneered intracellular profiling. So we took a single cell, we took an oocyte, so it's a large cell, and we put it in a cryostat, and we sliced it. And then we could measure the transcripts across the cell. And these are some digital PCR data showing the expression in a single cell from top to bottom. And you see that on the left slide, which is the typical distribution, most of the transcripts, they're shown in red in the digital PCR readout here, are more or less in the center of the cell. While the transcript to the right, Wnt11, you see that all the transcripts are actually in the bottom of the cell. They're very asymmetrically distributed. And that, of course, has consequences, because if you look at the bottom slide here, where we have the oocyte, and the arrow there indicates the direction of the gradient that we observed in the subcellular profiling study, you see that when the cell divides, the first cell division is like this, then the two daughter cells are still the same, because they are not affected or differentiated by the gradient. And the same thing was after the second cell division, so the four daughter cells here are still the same, but after the third cell division, then the top and bottom cells are different. So this is one example, at least, of asymmetric cell division. But the interesting thing is that the asymmetric cell division was there already, or prepared for, or programmed for, 3 cell divisions ahead when it actually occurred. So I think that was quite a cool concept. And as I said, this is 15 years ago. But anyhow, so what is the laboratory doing today? So we are very interested in healing and regeneration. And the reason for that is it's, of course, extremely important. We all know that when we as humans got a wound, at least as when we are adults, it heals quite poorly. It certainly does not regenerate. And in fact, if we today have kids that are, or fetuses really, that are born with certain diseases that require surgery, today we actually perform like heart surgery while the fetus is still in the wound, because it heals much better. So essentially our ability to heal, and particularly to heal without scar and regenerate, is lost over time. So we do have some ability when we are still in the womb. And there are certainly animals that can heal and regenerate. And of course, if we could learn more about those mechanisms, it will open up for totally new ways to treat wounds. And the other area, and this is the field of Radek Shindelka, and the other area is ischemia. That is essentially brain damage, where we also have very, very poor treatments. And now with the new techniques out there, single cell profiling and spatial transcriptomics, we are kind of learning more about them. So if you start with the Xenopus laevis, so Xenopus laevis, that's a tadpole, well, rather the tadpole of Xenopus laevis, which is the African claw frog. And the interesting thing about it is that if you cut its tail, it will actually regenerate. And when we do expression profiling, we can see that the regeneration is divided into different parts: an early response, then we have a healing, and then after the healing, the regeneration initiates. And if we just do a regular bulk expression profiling, we can clearly see these 3 different phases. That after the cutting, we have certain genes that are activated, and then the activity goes down. And then in the middle, we have another group of genes. You see the type of genes there that are activated. And then again, the activity is lost. But the reason that the tadpole is such an interesting model is that actually during development, the tadpole loses the ability to regenerate. And then it regains it again. So at cell stage known as 47, it can't regenerate. The tail does not regenerate. So something is different. And that makes it a fantastic model to really try to find out what's behind regeneration. Why can we regenerate under certain conditions and why can we not regenerate under other conditions? And so what we are comparing here is essentially the tadpole at two different stages. The stage where it can regenerate and the stage where it cannot regenerate. And it's very in time, it's a very short difference between them. And we see that there are totally different genes active in these, and the healing is a key phase for the regeneration that determines whether the tail can regenerate or not. And so we decided, I mean, so far it's just bulk data, so we decided to address that with single cell profiling using the Chromium system, the 10x system. I guess most of you are familiar with how it works. Essentially, we are isolating single cells with reagents in gems, and they are barcoded, and we get single cell expression profiles. So this is the workflow. We cut the tail of the tadpole, and then we collect the outer surface, and we do that at very, very short time intervals. That actually turned to be critical, that we actually, because things, we didn't know that took quite some time until we discovered that things actually happened quite soon after the cut. So our first data are just a few hours after the cut, and then we measure over a couple of days. And we do the QC of the data, we run the Chromium, and then we essentially analyze the data by cluster analysis. We look at the differential gene expression, and then we also model that by looking at the expression networks and ontologies. And doing that, so this is a new map clustering of the data. So what we found in this study is that after about 1 hour, a new group of cells, we call them RICs, that turn out to be totally determinant whether we get regeneration or not is formed. The RIC stands for Regeneration Initiating Cells. They're shown in green in this picture here. So you have the time resolution under A. I can point on that one. And in graph C, you see they are not present at all in the beginning, and then they increase, peaking around day 7 hours. And in the graph B, you can see how they develop over time. And D actually shows that if we use antisense technology, we can actually close down. Antisense technology to one of the marker genes that we later discovered is important for the RICs. We can actually close down regeneration. And the interesting thing about these RICs that we have now discovered is that they are actually evolutionary conserved. So it's not nothing that is specific for the tadpole, Xenopus levis. We see it in sturgeons, in rat, mouse liver, and it seems that everywhere we start looking at these, the RICs are very there, and they are extremely important whether we have an ability to heal a wound and regenerate, or if we will develop a scar and not degenerate. And the bottom panel here, that's immunohistochemistry for one of the markers reflecting the RICs. You can see that when we've just cut at 0 hours, we don't see the RICs there, they're not there at all. And after 1 hour, they start migrate, and after 6 hours, they're covering the cut. So the green fluorescence, that is where the RICs are. So we decided also to look at the phenomenon using spatial transcriptomics, using the VCM system. I guess most of you are familiar with that, but essentially we take a tissue slice and we hybridize it, and the spots on that are barcoded. So we have a spatial resolution. So when we do the sequencing, we can then trace back where on the tissue those transcripts actually originates. And these are some of the tissue slices that we collected. And here you have the spots that we are actually analyzing. This is now enlarged. And the RICs that we discovered, they are characterized by having a number of remodeling enzymes that are being active, and also TGF-beta inhibitors. So here we have directly on the spatial map, we have indicated where we see the TGF-beta inhibitors. They're shown in gray, and you can see them there in the bottom. And if we now compare the regenerating tadpole with the refractory, so the left one, the regenerating, they are healing, the refractory are not healing. And you can see very clearly that the TGF-beta inhibitors are only seen in the regenerating embryos or tadpoles and not the refractory. And if we close down the expression of the TGF inhibitors or some of the important remodeling genes using antisense technology, which then you can see the control is in the top, and the antisense animals in the bottom. You see the tail is regenerating in the control, but if we close down the expression in the RICs, we don't have regeneration. And it also seems that what is really refractory for regeneration, it's scar formation. So when we get a wound, either we regenerate or we get a scar. So that's the two alternatives. So what we see here in the top now, this is fibrotic staining for fibronectin. In the regeneration in the top, we see the bud coming out, one in the bottom, we see instead the scar formation. And again, if we take a model that usually regenerates and instead we close down TGF beta inhibitors or the remodeling genes using antisense technology, again, we get scar formation instead. So in summary, the end of the healing phase is the initiation step for the effective regeneration. And the regeneration initiating cells, the RICs, they express remodeling enzymes and TGF-beta inhibitors, and they are critical for our ability to heal. The second part is about ischemic brain injury, which is by Lukáš Valihrach. And essentially, we are talking about stroke here. So the critical reduction in blood flow in the brain caused by either sudden or gradual occlusion of cerebral arteries. And if we block blood flow, we get neurological deficits. And there are many pathologies caused by this. And this is important because it affects 12 million people every year, and that's the third leading cause of death and disability combined. So it's a major economic burden, and we don't really have any particular treatments. So although we have investigated a large number of drugs, we have not been really successful with that. So, and the reason it's very hard to develop a new therapy is really the complexity of the disease, and it involves so many different cells, and it's complex because of the development also over time and space. So, however, with these new omics methods, we can address that, and so that is what we want to do. So we have an experimental model where we essentially induce the injury, so we get blood clotting, and then we monitor that. We monitor that using 3-month-old mice, and we measure over time control 1 and 3 and 7 days post-injury. So we screened 5 mice per time point using magnetic resonance imaging and quantified the size of the brain damage. And the lesion volume was typically, well, it does vary a bit, but it's about 9 cubic millimeters. And on day 3, we saw it was more heterogeneous, but that's roughly how it looked. Yeah, I can actually do it like this. And then we started by doing a control, essentially having an area covering about 2,417 spots with a median getting about 25,000 transcripts and 6,000 genes captured per spot. And then using spatial transcriptomics, our pipeline, then we could iteratively place those and cluster those and identify regions in the brain. And this is essentially the healthy brain. And in total, we got 13 clusters, or we decided to go with 13 clusters for the brain anatomy as reference in the brain atlas. And then next, we started to compare the different regions of the brain, looking for differentially expressed genes. And in average, or typically, we found more than 100 differentially expressed genes between the regions that has a fold change of more than 1.5 and a Bonferroni adjusted p-value that is less than 0.01. That are essentially significant. You see that there. And we validated the data by comparing with the LM Brain Atlas, which is based on hybridization, in situ hybridization, and the graph to the right shows the correlations with excellent correlation with our spatial transcriptomics and the in situ hybridization. I can put it like this. So next we integrated the data for the control and for the ischemic sections to describe changes in the cortical gene expression that is introduced by the stroke, and we can then compare over time. And we got roughly 2,600 spots with a median again of roughly 12,000 transcripts and 4,000 genes captured per spot. And again, doing clustering revealed the cortical lesions areas, and they are separated based on, primarily based on time. So it's ischemia day, 1 day post ischemia, 2 day post ischemia, and 7 days post ischemia. Secondary, we have a separation between the central and peripheral location of the lesions. So, and then when we look at the data, while the lesion on day 1 showed very low gene expression, generally the activity increased with time. You can see that on the central slide. It peaked at day 7, but it never reached down to the lower level. So we never get a healing in these mice. So the healing actually starts, but they will never heal from this. And then, we essentially clustered the genes to get them. Get the ontologies in place and the networks that are being activated, again, separated over time where we have the largest changes. And then we can look at the data and see what happens over time and over space. And then generally, we see a persistent inflammation response, processes that are restoring the integrity of the damaged tissue. However, in contrast to early time points, we see broad classes of processes showing distinct spatial location, while reparative machinery is more prominent in the central areas of the wound. and inflammation takes place, primarily in the periphery of the damage or of the wound, and it involves the immune system, immune cell activation, and glial cell migration. So the changes that were introduced by the ischemic brain injury are orchestrated by multiple cell types. They complement the previous analysis with cell type specific content, and then we can apply computational deconvolution methods such that we can take the spatial data and correlate that with the single cell data and essentially resolve such that so we get a single cell or single cell type resolution in the spatial data, but it's a mathematical deconvolution. And this is how it looks for the deconvoluted data. So now we have the spatial transcriptomics, but resolved in different cell types. And this is 1 day post-injury. This is 3 day post-injury, so we can see where the different cell types appear and disappear. And this is 7 days after injury. And just to summarize, so the analysis, essentially summarized in the right picture there, uncovered dramatic changes in the cell type composition. So while the uninjured cortex was composed mainly of neurons, astrocytes, and oligodendrocytes, the typical cells in the brain, without injury, so when the injury caused a substantial decline in neurons, it triggered infiltration of the peripheral immune cells and proliferation of the glia cells. Vascular cells enter and fibroblast cells enter. And most changes were coordinated in time and space. We see neural death occurring starting on day 1, and we see it goes in a gradient from the central to the periphery. And it suggested that we have a gradual expansion of the injury. It peaks around day 3, with minor differences between the central and peripheral lesions. And on day 7, when the neuron count starts to increase a little bit in the periphery, it indicates that there is some kind of a neural reorganization. While the peripheral immune cells were not detected in the uninjured cortex, so we don't have any immune cells in the healthy brain, or at least we can't detect them, they suddenly become dominant in the lesion on day 1. And the largest contributions there, that's neutrophils, but we see also other peripheral immune cells like monocytes, dendritic cells, macrophages, T cells. Although some are, they are a little less abundant, but they are located to the space within the lesion. And glial cell proliferation is seen, and we see a differentiation also with other prominent processes. So there are major changes in the type of cells that appears, and the essentially the variance of the cells. So there are major changes, and that makes, of course, treatments very difficult. But anyhow, this is what I wanted to share with you today, showing that today we do have tools to essentially generate these spatiotemporal single-cell resolved maps, which are really fantastic to see what happens in a complex system like the brain. And I like to express major thanks to my colleagues, Radek Shindelka, who leads the group working with the Xenopus levis system and the healing, and Lukáš Valihrach, who leads the group working with the brain damage and ischemia. And I also want to mention that we work together with the Gene Core, which is a service laboratory that we set up 15 years ago, and that is totally focusing on single cells and spatial transcriptomics, and is essentially supporting academic work all across Europe. So if you are interested in getting help or support doing these kind of studies, just contact the Gene Core and they will be available. So thank you for attention. I should say that I hope that my colleagues are online to support me with questions, if you have questions, because I was not really responsible for this work. I jumped in because I happened to be in place. Well, thank you very much.
Thank you, Michael. With some questions?
Hi, fantastic talk. Thank you. Regarding the first part, so if do you think that what you see in the process of regeneration has something to do what we see, for example, in cancer, since TGF-beta is involved and it's also involved in the epithelial to mesenchymal transition, for example?
The answer is yes, because we have actually seen exactly these systems, cells, that seem to, if they are not bricks, they are very close to that. Yeah, and we think it's actually totally important for the growth of the tumor. Yes, but we don't know more than that. Yeah, it's very, very, very interesting question. Yes. Yeah.
Thank you for the nice presentation. I have a question regarding the second part. There are very few compounds that have been showing some benefits in that sort of situations. I was thinking to one mysterious preparation, a Chinese medicine preparation called Neuroed. It was, by the way, developed in my institute, or there was some work, and it showed some benefits. And I was wondering if you were planning to test this sort of compounds, because it would be interesting to document better what's going on, what are the cell types that are affected, and it could give some good clue about what could be effective in that pathologies.
No, this is an excellent question. This system is extremely complex, but it's certainly, the data gives some hints on what may work. And our team, or Lukáš Valihrach, well, actually, I see he is here. Yeah, if he can speak, then I think it's better he answers, because I know he has very exciting collaborations on exactly what you were saying. Please, Lukáš, can you take that?
Yeah, sure. Hello. So thank you for the question. So actually, could you hear me? I guess yes. Yeah. Actually, that's the plan with the data, that we would like to define some targets which could be somehow intervened with the different approaches. So currently, we are working on one approach using the C3A peptide, but of course this should be like data-driven discovery. So that's where we focus actually.
Other question? Okay. Thank you very much. We have a next speaker. Thank you, Michael. Great job.